Good morning, students. In the previous class, we have discussed about the structure of the chloroplast. Today, we'll discuss about the pigments involved in the process of photosynthesis as well as the absorption and action spectra. As the pigments, we know they are special type of molecules present within the plant body which helps to trap the solar energy. We can also observe within a single plant and even within the same leaf we can say different shades of the green we may observe within the leaves. So the reason behind that is due to the specific type of pigments present within their leaves, and we can easily separate out the leaf pigments of any green plant through the technique which is called as paper chromatography. So, a chromatographic separation of the leaf pigments shows that the color that we see in the leaves is not due to a single pigment but due to four pigments, which are chlorophyll A, which is bright or blue green in the chromatogram, the chlorophyll B, which shows yellow to green color, and the xanthophyll pigments, which shows the yellow color, as well as the carotenoids, which are, we can say, yellow to yellow orange color. So these pigments, they are the substances which are having the capability to absorb the light at specific wavelengths, and they help to carry out the process of photosynthesis. So, most specifically, the common chlorophyll pigment is chlorophyll A, which is the most important pigment playing vital role in the process of photosynthesis, and rest of these three, which are chlorophyll B, xanthophylls and carotenoids, these are the essential pigments, or we can say accessory pigments, which help to trap the light energy and help to transfer it to the chlorophyll A. And also we can say these accessory pigments, which are chlorophyll B, xanthophylls and carotenoids, they help or they prevent from photooxidation of the chlorophyll A pigment. So in these two diagrams we can observe that the figure, it shows the graph showing the ability of the chlorophyll A pigment to absorb lights of different wavelengths. This is the absorption spectra has been shown on the x-axis. The different light of different wavelengths have been put there in the sequence of VIBGYOR, that is violet, indigo, blue, green, yellow, orange and red. So here in this particular graph we may observe that the chlorophyll A pigment, which is shown in the blue color, it shows that it reaches a maximum peak at particular spectra and then it declines, and then again it reaches up to a certain height and shows maximum activity. Whereas for the chlorophyll B, we may observe the absorption spectra is somehow shifted right to the particular spectra shown by the chlorophyll A pigment, and whereas the carotenoids, which are shown by this yellow color line, it shows two different peaks and then afterwards it declines. So this particular spectra, this particular graph, it concludes that the chlorophyll A pigment is having maximum absorption capacity at two different wavelengths of light, which are in the region of blue region and the red region. In the B diagram, we may observe the graph, it depicts about the action spectra, which means the rate of the photosynthesis, which is measured by the O2 release. So obviously in which the specific wavelength the maximum absorption spectra is observed, in the same spectra the maximum action spectra would be observed. So this particular graph has been shown for the chlorophyll A pigment. So we may observe in the wavelength of that particular two different colors, which are blue and the red light areas, in these two different spectra, that the action is much more, or we can say the release of the oxygen is much more, and the rate of the photosynthesis is higher when the blue and the red light falls upon the plant body. In the third diagram, which is showing the graph showing the action spectra of photosynthesis superimposed on absorption spectra of chlorophyll A pigment. So here we may observe the absorption spectra, which is shown with the blue color line, and the action spectra, which is the rate of photosynthesis, is superimposed upon that particular, wherever the we can say the absorption is much more. So we can observe in these two different wavelengths of the light, the rate of action is much more. It is just, we can say, equal to that particular wavelength of light at which the rate of absorption is more. So at these two different points we may observe the peak. So in these two different points the rate of absorption is much more, as well as the rate of action is also much more in these two different wavelengths of light, and this shows that the, we can say, the action spectrum of photosynthesis superimposed on the absorption spectra of chlorophyll A pigment. Okay, and some part we may also conclude that the photosynthesis, it takes place, we have concluded here that it takes place in the blue and red regions of the spectrum, and some photosynthesis that do also takes place at the other wavelengths. At the other wavelengths, in the case of that particular different other accessory pigments do we have discussed of the visible spectrum, and now the chlorophyll is the major pigment responsible for trapping light, while other thylakoid pigments like chlorophyll B, xanthophylls and carotenoids, which are called the accessory pigments, they also absorb light and transfer the energy to the chlorophyll A. So these other pigments, which are chlorophyll B, xanthophylls and carotenoids, they are the accessory pigments, and they help to trap the light energy, and they finally transfer it to the chlorophyll A molecule to carry out the process of photosynthesis. And also it helps the chlorophyll A, which is the essential element pigment, to carry out the process of photosynthesis from the photooxidation. So till so far here, thank you, have a nice day.